Hello, welcome again to MLSQR Medical Lab Science Quick Review. And today, our interesting hematological topics are the thalassemias, including etiology, pathophysiology, lab findings, and question and answer revolving around the ASCP Board of Certification licensing exam. The word thalassemia was derived from the Greek word for sea, thalassa, and the stem haemia, meaning blood. Thalassemia replaced Mediterranean anemia in 1932 for the inherited anemia commonly found from ethnicities from the Mediterranean countries where malaria was also prevalent. Patients with thalassemia are common in a hospital setting where we run their CBCs and see their peripheral blood smears in hematology. And we cross match and prepare packed red blood cells in the blood bank for their blood transfusions. A quick review of hemoglobin and its synthesis will help us understand the thalassemias better. As you may be already familiar with hemoglobin A, the most common adult hemoglobin, there are two alpha and two beta globin chains plus four heme rings to make one hemoglobin molecule. The alpha and beta globin chains are quantitatively roughly equal. In beta thalassemia, we have a quantitative deficiency in beta globin chains. So we have a deficiency of the beta globin chains, hence the name beta thalassemia, and vice versa for the alpha thalassemia. Now, this is distinct from the hemoglobinopathies, where you have a qualitative defect or a structural change, a mutation that causes a structural change in the hemoglobin variant, and then also a RBC morphology change. Understanding some basic fundamentals of hemoglobin synthesis will help us understand more about the pathophysiology of thalassemias. Also, this relates to anemias, hemoglobinopathies, porphyrias, iron kinetics, red blood cell morphology, and more. Low oxygen levels in the blood or hypoxia stimulates tissues in the kidney to secrete erythropoietin, which will go to the bone marrow. Erythropoietin hormone stimulates the bone marrow erythroid precursors with their nuclei and mitochondria, and they are able to synthesize alpha and beta globin chains from chromosomes 16 and 11. Alpha like genes are in chromosome 16, and beta like genes are in chromosome 11. These include epsilon, gamma, delta, and beta genes. Let's quickly look at the alpha globin chain. Ferrochelatase is an enzyme in these erythroid precursors in the bone marrow to chelate iron in its ferrous state to protoporphyrin 9, and this will form heme, which will combine later with the alpha globin chain, forming heme plus alpha monomer. Likewise, the beta globin chain. Two alpha and two beta dimers come together to form a hemoglobin tetramer, the complete hemoglobin molecule alpha 2 beta 2, which is the predominant hemoglobin in adults, known as hemoglobin A. Hemoglobin A has a quaternary structure, which is the three-dimensional structure of the hemoglobin. Genetic defects in thalassemia include reduced or absent transcription of messenger RNA or messenger RNA processing errors or translational errors. This all results in reduced beta globin chain synthesis. If we don't have enough beta chains to make hemoglobin, then how does this affect the other hemoglobins? To understand the pathophysiology of thalassemias better, let's look at what happens in utero when hemoglobin synthesis occurs mainly in the fetal liver and spleen. Then the bone marrow takes over after birth. This graph shows that in utero, alpha and gamma chains are produced, with a decline of gamma chains after birth. Gamma chains are the ones used for hemoglobin F, the tough hemoglobin needed in utero. New beta chains rise rapidly after birth to replace gamma as the main non alpha globin chain forming hemoglobin A. And delta chains below rise slowly but are in the minority. These non alpha globin chains join with alpha globin chains to form hemoglobin A2. So now we see that in beta thalassemia major, hemoglobin A is absent. So in beta thalassemia major, hemoglobin F takes over, greater than 2%. In fact, it's above 90% in a lot of cases. Just a side note about hemoglobin F, we know that it's increased in beta thalassemia major, intermediate, and sometimes minor. But did you know it's also increased in hereditary persistence of hemoglobin F, in which the patients are asymptomatic? Hemoglobin A2 is variable in beta thalassemia, either normal or slightly increased. Excess alpha globin chains form hemoglobin precipitates, leading to inclusion bodies taken out by the reticuloendothelial system, red blood cell membrane damage, and ineffective erythropoiesis and iron kinetics ensue, followed by abnormal red blood cell morphology, hypo and polychromasia, and severe anemia. Massive expansion of the bone marrow tries to compensate. Clinical syndromes of beta thalassemia are based on the clinical severity of the anemia, whether they are transfusion dependent and genetics of the beta genes, if one or both genes are mutated, and to the extent of how these affected genes are expressed in quantity. Hemoglobin profiles with hemoglobin electrophoresis and high pressure liquid chromatography demonstrate hemoglobin A, hemoglobin F, hemoglobin A2, and hemoglobin lipor. Molecular genetics determine the mutations like beta 0 and beta silent genes, with advanced polymerase chain reaction and targeted mutation analysis helping with the diagnosis. Clinical presentation of children with beta thalassemia major include chipmunk faces with prominent foreheads and enlargement of their upper jaws. Skull x-rays often demonstrate a hair on end appearance. All of these are signs of bone marrow composition for the severe anemia from lack of beta globin chain synthesis. It's time to run the CBC and see the peripheral blood smear. This CBC report is consistent with a microcytic hypochromic anemia. Note that the hemoglobin, hematocrit, red blood cell count, MCV, and MCHC are all low. RDW is high due to the marked anisocytosis. Other microcytic hypochromic anemias include iron deficiency, anemia of chronic inflammation, and sideroblastic anemia, which includes lead poisoning. I noticed a tricky question in the ACP study guide that asks about interpretation of a similar CBC report like this one shown. And the RBC count was slightly high with microcytic red cells. What can it be? The answer was beta thalassemia minor. Let's look at the right game sustain of a peripheral blood smear of a patient with severe beta thalassemia major. Under high magnification, note marked anisopoikilo with many microcytes and macrocytes, many target cells, marked hypochromasia, polychromasia, nuclear red blood cells, see basophilic stippling and Howell Jolly bodies, and fragmented red blood cells. Hemoglobin gel electrophoresis shows normal controls in lanes 1 and 2 and beta thal major patients in 3 and 4, with AF and ASC controls in lanes 5 and 6 shown below. Note that the patient with beta thal major has an estimated 94% hemoglobin F. Hemoglobin A2 is variable. Precise quantitation is not possible, only estimations based on the optical density of the bands. For precise quantification of the hemoglobins, we need high pressure liquid chromatography. High pressure liquid chromatography offers a valuable screening test for beta thalassemia because of its high sensitivity and specificity. Quantitation of the hemoglobins is possible by integration of the areas underneath each peak. Molecular genetic testing definitely identifies the type of thalassemia and the specific mutation. Beta 0 means no beta globulin is produced. So beta 0 is homozygous and the most severe type of beta thalassemia. Beta plus means a reduced amount of beta globulin is produced. So it is not so severe when it's beta plus and beta plus. Delta beta Lepore is a less common type of beta thalassemia minor. Mentioned in the ASCP study guide, a question for MLS only. As a variant formed from the fusion of delta and beta genes during crossover in meiosis, forming a fusion gene product called hybrid delta globulins. And this combines with two alpha chains to form hemoglobin lipore. Let's quickly look at the alpha thalassemias. Alpha thalassemia silent carrier state has a deletion of one alpha gene. Alpha thalassemia minor has a deletion of two alpha genes. Hemoglobin H disease has a deletion of three alpha genes, and hemoglobin H is mentioned in the AACP study guide. This summarizes the genetics of the alpha thalassemias. One question in the AACP study guide for MLS only asked about hemoglobin H, found in hemoglobin H disease, also known as alpha thalassemia intermedia, more common in Asia. Hemoglobin H disease is caused by the deletion of three alpha genes, but there is one good alpha gene to make some alpha globin chains. Unfortunately, excess beta chains form tetramers known as hemoglobin H, which has a high oxygen affinity. And also know that hemoglobin H migrates as a fast hemoglobin in hemoglobin electrophoresis. Hemoglobin H migrates as a fast hemoglobin in hemoglobin electrophoresis, faster than hemoglobin A. By the way, not to digress, but remember the mnemonic for hemoglobin electrophoresis? The order of migration is AFSC, accelerate, fast, slow, crawl for the corresponding hemoglobins. So just remember, H is high speed, it's the fastest of all. Here we see an illustration of a peripheral smear stained 15 minutes with brilliant crescent blue, a supravital stain, which can demonstrate hemoglobin H inclusions, where you see violet blue punctate inclusions, giving a golf ball appearance. Let's go over some questions and answers that are similar to the ones presented in the ASCP study guide for the Board of Certification exams. Please like and subscribe and leave a comment. Thank you, and I hope to see you